Hi guys, I am going to explain photosynthesis. Let's see what is photosynthesis first. The process of synthesis of carbohydrate from carbon dioxide and water in the presence of sunlight by the green plants, and during this process they will release oxygen. If you want to see in depth, the reduction of carbon dioxide into a carbohydrate by water using the light energy. And where it occurs? It occurs in the chloroplast of green plants and in some microorganisms like bacteria. Now let's see the chloroplast. Chloroplast is a double membrane bound organelle. It contains membrane system called grana, which is stalks of thylakoid. It has stromal lamellae which connects the granas, and the matrix is called stroma. It also contains some starch droplets and lipid droplets. Now see the pigments in the chloroplast. Chloroplast contain chlorophyll A, chlorophyll B, xanthophyll, carotenoids. The colors mentioned by the side of these pigments are the colors seen when they are extracted from the leaves using the chromatographic method. These pigments are embedded in grana membrane. These pigments can absorb light at specific wavelength. Now look at the absorption spectrum of different pigments and the wavelength at which the rate of photosynthesis is maximum. You can see the wavelength at which maximum photosynthesis occur and the wavelength at which maximum absorption of light by chlorophyll A is similar. So it is chief pigment. Others are accessory pigments. These pigments are arranged to form photosystem 1 and photosystem 2, which is located in thylakoid membrane. These photosystem 1 and 2 are named on their sequence of discovery. That means the photosystem 1 is discovered first and photosystem 2 is discovered second. Photosystems are made up of two parts, antennae and reaction center. Antennae is formed by accessory pigments. Reaction center is formed by chlorophyll A. Based on the maximum wavelength absorbed by chlorophyll A in photosystem 1 and 2, they are named as P700 and P680 respectively, because chlorophyll A in PS1 can absorb light at maximum wavelength of 700 nanometer, and in PS2 it can absorb maximum wavelength of 680 nanometer only. Accessory pigments protect the chief pigments from photooxidation. Now what is photooxidation? It's nothing but when there is a high light intensity, chlorophyll pigment can damage and photosynthesis will not occur. So the accessory pigments protect these chlorophyll pigments from photooxidation. Now what is the use of photosystem? It helps in photophosphorylation. Now what is photophosphorylation? It is nothing but synthesis of ATP from ADP and inorganic phosphate in the presence of sunlight. It means the addition of phosphate group to ADP to produce ATP. This photophosphorylation also there are two types, non-cyclic photophosphorylation and cyclic photophosphorylation. Let us see it detailed. Non-cyclic photophosphorylation occurs in thylakoid membrane and cyclic photophosphorylation occurs in stromal lamellae, because PS1 and 2 both are present in thylakoid membrane, but in stromal lamellae only PS1 is present. In thylakoid membrane, the arrangement of PS1 and 2 is this. They will have an electron transport system in between them. The electron transport system consists of plastoquinone, cytochrome B6F, plastocyanin. Now we covered the basic things which you need to know before understanding the process of photosynthesis. Now we move into the reactions which involved in the photosynthesis. It occurs in two major steps. First one, the light reaction, and the next dark reaction or Kelvin cycle. Light reaction occurs in the thylakoid membrane because pigments are located over there, and the dark reaction occurs in stroma. Dark reaction, it doesn't mean that it will occur only in the absence of light. It is light independent reaction, but it depends on the products of light reaction. Let us see first the light reaction. The light reaction is the phase where energy and electron donor, that is ATP and NADPH, are produced using sunlight. Later, these are used up by the dark reaction to convert our carbon dioxide to carbohydrate. Light reaction is a combination of photophosphorylation and chemiosmotic hypothesis. I will explain first photophosphorylation. Look at the picture. In PS2, the base is reaction center and remaining part is antennae. In the reaction center, the water molecule is split into oxygen and hydrogen ion and it releases electrons. Using light of 680 nanometer wavelength absorbed by chlorophyll A, which is a reaction center, and the electron released in this process is excited and jump into higher energy level. In this higher energy level, the electron is taken up by electron acceptors and pass the electrons through electron transport system. When these electrons pass through electron transport system, the electron loses energy and phosphorylate an ATP during this redox reaction. They will also transport hydrogen ion from the stroma into inside the thylakoid membrane. Simultaneously, the electrons in PS1 also excited when the chlorophyll A in PS1 absorbs red light of wavelength 700 nanometer. This light excited the electrons to the higher energy level. There, the electron acceptor take these electrons and they reduce the NADP plus in stroma, which is present in the stroma, into an NADPH using an enzyme called NADP reductase, which is located in the stroma. You can see the electron moved in a Z shape from PS2 to PS1. So this non-cyclic photophosphorylation is also called Z-scheme photophosphorylation. In cyclic photophosphorylation, only PS1 will work and the excited electron will return back to the reaction center of PS1 through electron transport system. Cyclic photophosphorylation occurs only in stromal lamellae because there is absence of PS2 and NADP reductase enzyme, and it occurs when only the light of wavelength beyond 680 nanometer are available for excitation. Now let us see how the light reaction works by the combination of chemiosmotic hypothesis and photophosphorylation. Chemiosmotic hypothesis works on proton gradient created by photophosphorylation and it will produce ATP. What is proton gradient? Is nothing but increased concentration of hydrogen ion across the membrane. Now, how this proton gradient is created? In photophosphorylation, while splitting of water molecule in PS2 inside the thylakoid membrane, it will release hydrogen ion inside the thylakoid membrane. This will increase the hydrogen ion concentration inside the thylakoid membrane. This first process. Next, when the electron pass through electron transport system from PS2 to PS1, it will replace an hydrogen ion from the stroma to inside the thylakoid membrane. And the next step, when the electron from PS1 reduce NADP plus to NADPH using NADP reductase enzyme, They used the hydrogen ion present in the stroma that causes decrease of hydrogen ion concentration in stroma, and compared to stroma, inside the membrane the hydrogen ion concentration will increase. By these three process, the hydrogen ion concentration inside thylakoid membrane is increased when compared to the stroma by the process photophosphorylation. I told you that chemiosmotic hypothesis will generate ATP using the proton gradient. Now let's see how it's generate ATP. The increased proton concentration inside the thylakoid membrane increase the acidity of thylakoid membrane. To neutralize that, it have to push out the hydrogen ion to the stroma. There is only one way for the hydrogen ion to go out of the thylakoid membrane, that is ATP synthase, which consists of two parts, CF0 and CF1. What is CF0? It acts as a channel across the membrane. CF1 is an enzyme, helps to produce ATP using the energy released during the breakdown of proton gradient. When the proton passes through the ATP synthase, it releases energy. This is the breakdown of proton gradient. Using the energy, CF1 will add an inorganic phosphate to an ADP to produce an ATP. This is the energy producing center in the chemiosmotic hypothesis. We have done with the light reaction. The byproducts of light reaction is ATP, NADPH and oxygen. The oxygen is released out of the cell. The remaining ATP and NADPH will be used up in the dark reaction. Let us move on to dark reaction, that is Kelvin cycle. The scientist Kelvin found this process, so it is named after him. Now we are going to produce glucose from carbon dioxide using the byproducts from the light reaction. The Kelvin cycle occurs in four major steps. First, carboxylation, reduction, carbohydrate formation, regeneration. Carboxylation. From the name carboxylation, we came to know that carbon dioxide is added. Now the question, carbon dioxide is added to what? The primary acceptor of carbon dioxide is RUBP, ribulose biphosphate. It is a 5-carbon compound. Now in this process, the carbon dioxide combined with the 5-carbon compound ribulose bisphosphate, and it will produce 2 molecules of 3-phosphoglyceride, which is a 3-carbon compound. For the catalysis of this reaction, the enzyme is Rubisco, RUBP carboxylase oxygenase, and water is also used up in this process. The next step is reduction. The 2 molecules of 3-phosphoglyceride is converted into 2-glyceraldehyde 3-phosphate or triose phosphate. In this reaction, 2 ATP and 2 NADPH molecules are used. This is a reduction process. You can see that addition of hydrogen from NADPH to the 3-phosphoglyceride to reduce it to glyceraldehyde 3-phosphate. The next step is carbohydrate formation. One glyceraldehyde 3-phosphate is converted into a carbohydrate, and then another glyceraldehyde 3-phosphate is used for the regeneration process. The next process is regeneration. In regeneration, glyceraldehyde 3-phosphate is again converted into RUBP using an ATP from light reaction. Like this, the cycle will continue. We know that glucose is 6-carbon compound. Now we saw the cycle which fixed only 1 carbon dioxide. This means the cycle have to work 6 times to make a single glucose molecule. For fixation of 1 carbon dioxide, it used up 3 ATP and 2 NADPH. So totally, to produce 1 glucose, it needs 18 ATP and 12 NADPH from the light reaction. Finally, we synthesize the glucose. Is it we? No, we can't synthesize the glucose. Only the green plants synthesize the glucose because we doesn't have the pigment and enzymes in our skin to produce the glucose from photosynthesis. But ultimately, every living organisms gain their energy from the primary producers, that is plants. So the ultimate source of energy is photosynthesis.